Welcome to Where is Truth? We're so glad that you could join us. My name is Norman Peckham, and I have with me Dr. Ariel Roth, scientist, author, educator. I believe one of your first books is in now 16 different languages.
That's right.
And the more recent one in 8 languages and three more translations shortly.
Right.
We're so privileged to have you with us. We're going to talk about a very perplexing question for those who believe in evolution. For the creationists, it's simple. Where did life come from? God created it. But for the evolutionist who doesn't believe in God, it's a different story. It's relatively easy for us to tell dead things like mountains and rocks and the like from the beautiful green grasses and trees and flowers. But where is the border between life? How small can it get between life and death?
Well, we're addressing your very important question, and one that is significant because life is so meaningful and so important that you cannot bypass this question as to where life came from in this great battle between evolution and the Bible. And as you mentioned, we can tell easily living things from non-living, except there's some things that are marginal. A dead fish can be marginal because we don't know how dead it is. But it comes to viruses. Some people think, viruses, maybe life first started as viruses because they're simpler than ordinary life. Mm-hmm. But viruses do not qualify as life at all. For life, you have to have cells, you have to have reproduction, you have to have metabolism. Viruses have none of that. And they're reproduced by the cells in which they are and so on. So we can discard that, but there are some very small organisms that are at least simpler than we are.
Okay.
We're probably familiar with Escherichia coli. We hear about it quite often in the news and so on. Here's a picture of it. It's a very teeny microbe. 500 of them end to end for 1 millimeter. It tells you how very small this is. But when we look at this in detail, and the reason we're talking about Escherichia coli is because we know so much about it, we find it is an extremely complex thing. For instance, here's a list of the composition of one single Escherichia coli organism. This is just one all by itself. It has over 2 million protein molecules, and it has over 4,000 different kinds of protein molecules. And then you go down this list and you can see all kinds of other things. The thing is extremely complex. How did this all come about is the question we're addressing here as we look at this particular issue. Mm-hmm. And of course, one of the important questions is DNA. And we've all heard DNA and so on. Here's a picture of DNA, and of course Escherichia coli has DNA. When you look at this picture, you notice in the middle of the right-hand portion, which is an enlargement of the left-hand portion, you notice the letters A and T, TA, CG, GC, and so on. Those are the all-important genetic code. This is what make organisms what they are and how they work and so on. And we have a lot of these units. You have just a teeny little portion here. Escherichia coli has 4 million bases in its genetic makeup.
Each one of those letters is a base?
Is a base.
Okay.
We've got 3 billion of those in almost all of our cells. So that, this is very extremely complex. how did this happen? Could it happen just on an Earth that's empty? And as scientists who believe in evolution try to suggest, well, it could come together this way and that way and so on. This is the question we're asking. Is there a God or that designed this, or did it happen all by itself? Simpler molecules, but extremely important, are proteins. As we mentioned, we have over 4,000 different kinds of proteins in Escherichia coli and so on. Proteins are made of amino acids. In this figure, amino acids are represented by a little circle, and you got 100 of them there in order, and they attach to each other in a chain, and then they fold up and form a protein molecule. And those protein molecules are marvelous things that do all kinds of things.
It's interesting when they fold up. So they fold up in such a way that they might even have little robotic arms on them and do mechanical things.
Well, it's incredible what these protein molecules can do, you know. And then some of them will squeeze so that they don't work anymore and open up when they need to work. And you've got some forming coils and so on, chaperones that determine there's a great deal of complexity far beyond what you're seeing right here in this simple diagram, but they fold up and work and produce these various things. And just to, if you wonder about how amino acids are and so on, the chemical composition structure is shown in this particular diagram. And the black part, the chemicals that you see there and so on, that's constant for amino acids. The red part changes. Some amino acids are very simple, like the one at the left. All it has is hydrogen there. Others have a complex cyclic pattern. This is what makes the different amino acids, is the differences in that red part. But how do proteins get put together? And there's a marvelous machinery that does this, and we call them ribosomes. Now, in Escherichia coli, we've got 20,000 ribosomes putting proteins together here, and they put the amino acids together and so on. You got a diagram here that tells you a little bit about how they do this. You see the red dots? Those are amino acids. Mm-hmm. And they come towards that long blue line you have down there, which determines what kind of amino acid is selected and so on. And then this all goes into that ribosome and it's put together. And on the left side of the ribosome, just a little above the middle, you notice a red line going out. That's the protein. And those amino acids are all there in the right order as dictated by the genetic code that's on that big blue line there a little lower down. Scientists have been spending a lot of time, certain of them anyway, trying to determine, how could life arise by itself? Because at present it's not popular in science to think about God. You like to think about things just happening by themselves, mm-hmm. and so on, according to the laws that we've been able to determine and so on. And a landmark experiment was done at the University of Chicago. It's called the Millerurey Experiment, and you have it pictured here. And the important part is that big whitish, roundish bulb that you see towards the right there. You have various gases in there, and you have sparks. And they were able to make a lot of amino acids.
Are the gases that were in that vase, are they the kind of gases that we have now?
Somewhat, but absolutely no oxygen. You cannot have oxygen here because oxygen is destructive to these delicate molecules.
Do we know a time in history when there was no oxygen?
Well, some scientists suggest there was no oxygen when life originated, because if it was oxygen there, you couldn't have had it. So
oxygen would have destroyed this experiment.
Absolutely. But there's a lot of argument about this. A lot of data suggests there was oxygen. Some say, well, no, this data suggests there wasn't, and so on. It's a much discussed point.
Well, are there alternatives to this kind of process?
Yes, because this is not considered that satisfactory, although the millions of students in biology are subjected to this diagram here, and they're told, look, we can make amino acids in the laboratory, therefore life originated by itself. Mm. And That second part is the
Seems
one
like a
that's
big
under
move, a
discussion.
big jump.
It's quite a move and so on. But when you look at alternatives, and there are many of them, we don't have time to consider them. One of the main ones they're suggesting nowadays is that RNA was involved. RNA has certain characteristics, maybe a little bit easier in connection with reproduction and so on. But RNA molecules are extremely hard to make in a laboratory. You can make all the RNA you want to. You do not have the information that you need for getting the right amino acids to produce the right kind of protein so you have life.
The DNA has to be there with the information.
Absolutely.
So that the RNA can be proper.
Yes, they start with RNA, however, but RNA doesn't have the information. Let me tell you just a little bit about some of the problems we have with going beyond the amino acids, the origin of life, and so on. The more soup you have, the greater the chance you have of this happening. Now, the soup is what's considered, it's called the primordial soup. And so you need a lot of soup, so why not have all the oceans be the soup?
Okay.
And it's be somewhat like broth, full of amino acids, and so on. And this is the general, the most popular proposal. But when you look in the rocks, you don't see any evidence of that soup. If you had this broth around there, in the old rocks, and here's a picture of the Grand Canyon here, and you've got younger rocks towards the top, older rocks towards the bottom. You go below, and see the river there in the middle of it. You go below that river and you get into those older rocks where life is supposed to have originated.
No soup?
No soup. No evidence of organic material. I mean, it's not. So you can talk about this soup, you can talk about these amino acids. It's not there.
No evidence for it.
No evidence for it, at least, that you'd expect. A second problem is that you have to select the right amino acids. Now, when you make amino acids in sparks, like suggested, and this is supposed to have happened in the ancient world, it's lightning and air in the atmosphere and so on, making amino acids with these gases. You make more amino acids that aren't the ones that animals and plants use than those that they do use.
How many amino acids
20.
are there?
20 amino acids.
In the human.
One minor exception.
Yeah.
Yeah. In all of us, all living things.
But then how many do those 20, are they a subset of a much larger group?
Yes, because you have all kinds of different kinds of amino acids beyond that. How did the first life, every time it made a protein, select only
those 20?
Those 20. And then you got another selection problem, and that is because these amino acids come in two forms. They're just like our hands. You know, we got our right hand and our left hand. Look at this diagram right here. On the left side, we have that's what they call the left-handed, so to speak. It's the way it changes polarized light. It's not based on hands at all. On the right is the dextral, which is the right-hand type.
One looks kind of like the mirror image of the other.
You see, one's the mirror image of the other. But in living things, almost exclusively, we have the L type. And
that's
how
random.
So when you were making this first life, you don't have to pick the right kind of amino acid, but you got to pick the L type of that type of amino acid, because when you make these by lightning and so on, you have about half and half of each. Mm-hmm. Then there's a question of organic molecules would not survive. Calculations have been made regarding, when you have this atmosphere, 97% of the simplest amino acids, and they're the least delicate, would not survive through that atmosphere because of lightning and ultraviolet light and so on.
So even if you could make it by chance, it wouldn't last.
And then they figure 3% that's in the soup, they disappear. You have to get all these amino acids in one place at one time, all the L type,
right order.
the 20 type only you have in the right order to get life started.
Well, so what are the chances of that happening? Has anybody calculated that?
Yes, this has been calculated, and supposing you're going to make a protein, there's one chance out of 20 you're going to get the first amino acid,
right?
Correct. Second one chance out of 20 that you'll get that one right. For you to get both of them right is one chance out of 400. Because in probabilities, you multiply them and so on.
So now if I have 100 amino acids that I got to get right.
Now, this slide shows you the calculations of 100 amino acids. You want to get them right? You get the L types, you get other factors put in there, and so on. What is the chance that you might get that protein? It's not 1 out of 10, not 100, and so on. It's 1 out of the number 49, followed by 190 zeros.
Now, don't mathematicians have a kind of an idea that 10 to around 50 is the limit of what's possible?
10 to the 50th power, yes.
Anything more than that is impossible? And here's to the 190.
190, yes, yeah. You see, it tells you how, when you have such impossibilities, it's time for you to look at some alternatives.
Absolutely.
Then there's the genetic code. How did that come about? You see, in this DNA, when it dictates for a certain amino acid, it uses 3 bases, and those 3 bases dictate which amino acid will be selected to make the right protein so you can have life, type of thing. And here's simply a picture of that genetic code. It really, it's a little complex, but it's...
but I guess that's the point. It's complex.
Exactly. You have 64 possibilities and so on, and some amino acids have more than one combination type of thing, but everything is taken care of. You have a stop and a start code and so on and so forth. But we come back to this picture of the ribosome, which tells us a little bit about this genetic code and what's involved here. In that picture, you notice in the middle towards the right 2 bluish areas. Those are called transfer RNA. Those have a genetic code on them. And that genetic code matches itself to the genetic code on the long blue line below.
On the DNA.
And that genetic code dictates the order of the amino acids so you have the right protein. You can have some substitution proteins, but very little. So it has to be quite rigorously followed. And when the amino acid attaches itself to that transfer RNA, there's a special molecule that does that. So you have to have a specific amino acid. You have to have a special molecule that attaches that to it. You have to have that transfer RNA with the right genetic code. And then it attaches itself to this, and then you can get proteins.
All to get a protein.
All to get a protein.
And you have to have them all to have life.
Yes, at least 100 for a protein, that's a minimal figure. Some substitution, not very much, is allowed in this, and so on. Well, then we have biochemical pathways. What are these? We've just been talking about one little step. In producing some of these complex molecules in our body or in Escherichia coli and so on, it takes sometimes many steps. And these are illustrated by the rectangles at the lower part of this picture here, where A is changed to B, to C, to D, and so on, and G is what you want. In order to change A to B, you use an enzyme, and enzyme A and the ovals above does that, and the C changes C to D, and so on. Now, how did these complex systems get started? And keep in mind, in a number of cases here, you don't have any survival value, because until you get G, why should the rest evolve?
Right, there's no reason.
So, very complex systems that we have to account for in this. And then, how did cells form? The cell is a very complex thing. We're talking about chemicals here and so on, but get to a living cell, it is very complex. And evolutionary biochemist, Harold Moritz, decided to use thermodynamics, and that's the energy relationship of molecules, to try and determine, hey, what's the chance that the smallest organism we know that's free living, which is mycoplasma. It's got 482 genes. It's got about 500,000 bases in its DNA and so on. What's the chances that this could come together? And his calculation is here represented on this slide. It's 1 chance out of 10 to the 5 billionth power. Now remember, each one of these zeros that you add in this 5 billion multiplies the improbability by 10.
It kind of reminds me of if you had 1,000 monkeys jumping on 1,000 typewriters, what is the chances of getting the Declaration of Independence?
Yeah.
Infinitely more possible than this.
Right. And here's a picture of an animal cell just to show you a little bit all the different parts we have in that animal cell. I mean, you have to have this. I wouldn't say every one of those parts is necessary, but you have to have an awful lot of necessary parts before you can have life. And then you have the question of reproduction. How did reproduction start? You can have a little cell here, you know, of these chemical proteins here, and they got so on, and it's living. That is not going to start life on Earth by itself.
Because before long it'll be dead.
It's going to die, and that's the end of it. You have to have reproduction. And in reproduction, you have to reproduce all those essential parts, or you're not going to have life starting on the Earth. How did reproduction get started? This is a very basic question that is not answered along with the other problems we're suggesting. So on picture here of reproduction, this is in more advanced cells a little bit than Escherichia coli, but it tells you how complex that reproduction process can be, where you reproduce your DNA, all your other essential parts have to be reproduced. It's the ordinary process of mitosis that most people have heard about or studied about in their basic science courses type thing. Could this all just happen by itself? You know, it does not seem possible that life could arise by itself here on Earth, considering all these special characteristics that we have for life. And then lastly, I mentioned the origin of the DNA proofreading and editing system. Now this is something you may not have heard of. When you reproduce organisms, you have to reproduce the DNA.
Yes.
That's the very important part.
Right.
Now, when you reproduce DNA in the laboratory, using the various enzymes and so on necessary, you get about 1% error. This is totally incompatible with life. Life, we have to have precise labels. The proteins have to be usually very precise, very close at least to a certain pattern type of thing. But there is a wonderful system which essentially reads and edits the DNA to make sure it's copied properly. And if there's a mistake, it removes the mistake and puts in the proper base so that the DNA is correct.
So where did that intelligence come from?
Well, and this raises a very basic question here. When life arose, which arose first? The DNA editing and proofreading and editing system, or the DNA? You have to have both. Without both systems, reproduction of DNA and the proofreading and editing system, you cannot have life. Well, let me tell you a story. In December 9, year 2004, Associated Press released a story that Antony Flew had decided there must be a God. Now, most people don't know Antony Flew. He's probably the most notorious scholarly atheist that there has been. He's devoted his life. He's written over 24 books, largely advocating atheism. He's been the icon for intellectuals in the atheist ideas. And all of a sudden he said, there's got to be a God.
What made him decide that?
On the basis of what we've been talking about here. DNA. He says that Charles Darwin, Richard Dawkins, and so on, they totally ignore reproduction. You've got to have reproduction type of thing. He's also impressed by the precision for the Big Bang, forces of physics, that type of thing. It's the science that convinced him there's got to be a God. And when you look at all these details, I think there's got to be a God that did this.
Well, it's an amazing story. With the complexities, the statistical analysis that has been brought to it, that the mathematicians say it's impossible. It's only the life science biologists who cling to this notion of evolution. Most of the other scientists aren't interested in it and are not so sure about it.
Not as strongly.
That's right. With all these evidences and the tremendous complexity that we have here, it seems like it takes as much or more faith to believe in evolution as it does in the simple story of creation. We'll see you next time in the search for Where is Truth. If you would like additional free information about these discussions, there is a PowerPoint resource available on the internet that covers many more details than could be provided in this lecture series. The resource is titled The Bible and Science and is available at scienceandscriptures.com. Be sure to add an S at the end of the word scriptures to get to the right web page. The Bible and Science resource follows the same sequence of topics as the Where is Truth series, but it is divided into 17 separate discussions instead of the 14 videos of the Where is Truth series. The title of the discussions, an introductory outline, and an index will lead you to the topics you want. The Bible and Science discussions served as the resource for the Where is Truth videos. To facilitate understanding and learning, each of the 17 PowerPoint discussions in the Bible and Science resource is provided with a set of questions and their answers. You will find these at the end of each PowerPoint presentation. The Bible and Science resource was designed especially for students and teachers. However, it can be of great help to anybody interested in the topics.